we can start.
Yeah, please, no problem.
Yeah, thank you.
A very good afternoon to one and all. I would like to take this opportunity to express our hearty welcome to our speaker for this session, Dr. Deepak Kumar sir. Welcome, sir.
Yeah, thank you.
Thank you, sir. Dr. Deepak sir holds PhD in biotechnology and he is presently working as senior scientist at Ella Foundation, Hyderabad. Sir is veterinarian and virologist with more than 15 years of research experience. Sir has involved in 3 pandemic vaccine development projects such as influenza, H1N1, Ebola, and COVID-19. Sir's wide experience involves in vaccine development. The 2 main vaccine development for COVID-19. One is inactivated SARS-CoV-2 vaccine, which is very well known internationally as Covaxin. Second one is Japanese adenovirus 36 vector SARS-CoV-2 vaccine, which is PBV154. Such other interest areas and vaccine development areas involves Ebola virus disease, human adenovirus 5 vector Ebola virus vaccine development, as well as in swine flu department, inactivated influenza AC California vaccine HNVAC. Sir has several publications and 2 patents, namely coronavirus vaccine and method for preparation of thereof, as well as coronavirus vaccine through nasal immunization. We are very lucky to have such a distinguished personality with us today. Now I request Dr. Deepak sir for his talk on roadmap for viral vaccine development. Please, sir.
Yeah, thank you. So can I share the slide
please.
now?
Yes, sir, please.
Yeah. So is it visible?
Not yet, sir.
One minute. I'll close my video. Yeah, is it visible now?
Yes, sir, it's visible now.
Yeah, I'll put it in. Yeah, is it in full screen? Can I go ahead?
Perfectly visible, sir. Please.
Yeah. So good afternoon to everybody, and I'm glad that I was contacted for this presentation. So today I would like to walk through the various steps that are there in regulatory, including regulatory approval for viral vaccine development. And probably I'll just give you a gist of what happens in a vaccine industry. So what are the different departments that are involved, and what are their function, and how do they coordinate to get the vaccine out. So so we know that this is the list of some of the well-known vaccines that are that everybody is aware of. So to this list, I think now adenovirus vector vaccine as well as the mRNA vaccine are the new candidates which has added to this list. But coming to the question of what is an ideal vaccine. So an ideal vaccine, if we talk about that, it should produce immunity which is similar to what we get from natural infection, without causing any adverse events or adverse effect or any illness. So that is the best vaccine, and it takes a lot of time for developing such vaccine. But this image looks simple, but that is not the case. So if we look at our vaccine simple, if we ask this question, it's not 1 + 2 = 3. It is a lot more than that. As simple, it looks simple. What I did is I put this virus plus cells and you get the vaccine. This is the simplest way we can explain it, but that is not true. So moving ahead, the regulatory roadmap for vaccine development. So there are a lot of things which are regulated when we go for vaccine development. So initially we do R&D studies, and then we think about going ahead for the vaccine. Later, we need to do preclinical toxicity study to show that it is not toxic to the animals. The 3 different animals need to be used, especially one rodent and non-rodent is enough for doing that. And after proving that it is not toxic in animals, then comes the next part. We move ahead for approval and this thing, clinical studies. But all these things are we need a lot of approvals, regulatory approvals, and it's under a big umbrella. So each and every aspect is taken care of. Different committees which are there in place. So we have Institutional Biosafety Committee, IBSC, Animal Ethics Committee, RCGM, which is the DBT body, and CDSCO. You must be aware of this. So these are all Drug Controller General, and these DSMB. They look into the toxicity in the clinical trial phase 1 study, and the SEC committee, you may be hearing a lot about them as subject expert committee. So they review the clinical trial data, and then decide or pass on pass on their recommendations to the CDSCO. And NTAGI is another body which is independent, and they actually look into how to include the vaccine in the schedule and how to be given. So they are again, that is again another regulatory body. So this is when we are talking about R&D, preclinical and clinical. But is there any regulatory regulation that goes on when we produce the vaccine? So to answer that question, let us take a look on how what are the regulations that are there during vaccine production. So if we talk about vaccine production, I have put it very grossly here. So production, then followed by formulation, filling, batch release, and transport to the place where it needs to go. But all these things are monitored. It's not like an independent event, and all departments they are doing their things independently. All these are coordinated, and quality control department tests each and every sample which is coming from these departments, and it is monitored by quality assurance team. And again later the NRA, National Regulatory Authority comes into the picture. They test samples, and when they say okay, it goes for batch release. So let us now zoom into what happens in individual this thing in production. So production process we can categorize into upstream process and develop the downstream process. So if we look into upstream process, we need usually for this thing viral vaccines, we need a characterized cell, bank cell, which is like master cell bank and working cell bank need to be prepared, and everything is documented. So the characterization of the cell line is very important, and the characterization of the virus that is being used is also very important. So when we look at upstream process, I have tried to put it in as simple as possible. You can see that the cells can be infected by virus, incubate it, and later harvest it. Up to this part, it is upstream process. So when we look into microscope in small scale, it will look something like this in flasks. So you can see here uninfected cells, and these are infected cells. But what happens in cell stacks and all, these are larger stacks, so we cannot see actually what is happening. But all backup data need to come from R&D for supporting how much virus is required and to infect the cells. So there is another thing that is being used for bioreactors, the adherent cells which stick to the surface of the stack or the flask. If we want to grow it in suspension, it is difficult. We need to adapt, and in that process, a lot of passages happen. So that may change the nature of the cell. So for that purpose, there are microcarriers available. which can be used, which is of definite shape, and cells adhere to that. So these are sorry. So these are the microcarriers that you can see here, these round objects in the image. So cells are seeded. Initially, they will look round, and then slowly they start growing on the surface of the microcarriers. When these are infected, here in the image D that you can see here, so when these are infected, you can see that slowly the cells are dying, and you can see that the microcarrier is again kind of empty. So these are this can also be used. But the question is, like, you probably can think of this, what can QC and QA do here? So there are a lot of things that can be done here. They can check for sterility. They can check for whether the beads that were received, whether they have a COA for that. So a lot of things can be done here by QA and QC. Sorry. So coming to the next part, upstream process. So, in this, so substrate, we can, in the upstream, we can consider substrate as cells. Right now, these are the cells that are being used for vaccine production. Vero cells, it's African green monkey kidney origin cells that is used for most of the vaccine. And MDCK cells are originated from canines. This is made in Derby canine kidney cells. And HEK293 is currently being used for adenoviruses. And BHK cells are bovine hamster kidney cells. These are used mostly for animal vaccine production. So if we talk about viruses, again, the best candidate need to be selected based on R&D or preclinical studies. And propagation, how much virus to add to the cells. This is called as optimum MOI, so multiplicity of infection. This need to be determined for each vaccine that is being used. So coming to the next part, harvest. This again, we need to find out a proper time to harvest, which again need to be determined for each of the vaccines. So QA and QC, they do a lot of things and keep document of each and every aspect that is every research that is carried out at different departments, and this support the regulatory requirement for the particular vaccine. Coming to the next part, the downstream process. Again, I have tried to put it in as simple as possible. So harvest, there are a lot of things that can be done with the harvest. So initial clarification is important to get rid of the cell debris, and followed by purification, concentration. If it is inactivated, inactivation is done. So these are used in different combinations to get the drug substance. That is, I have mentioned it as DS here. Later, which based on the formulation type, this again, this is formulation is guided by the R&D studies. So formulation, after formulation of drug substance, the required amount of antigen is used, and the drug product is formed. So this drug product, so this is just an example of how this purification or this thing buffer action can be done with the TFF column. So you can see here smaller molecules go out, and the larger molecules which contain virus particles can be retained, and that can be concentrated further. So QA and QC, they can again do a lot of things here. They can look at the antigen loss. If at all there is some investigation, QA can step in and see what is happening. And they also aid in determining the antigen content in the drug substance and the drug product. So coming to the in little bit detail in downstream process. So inactivation, lot of inactivating agents are available, but these are the mostly used beta propanolactone, vinylethionamine, and this thing formaldehyde. So BEI and formaldehyde. So these are mostly used for vaccine production. BEI is mostly used for veterinary vaccines, for FMD vaccines. BPL and formaldehyde in human vaccines, it is used. So if it is a live vaccine, some or a vector vaccine like adeno, inactivation is not required. But if it is a kill vaccine, inactivation is required. And nowadays there are UV based method of inactivation. All those things are coming up, but still it will take some time to come into the mainstream. And regulatory need to accept those kind of inactivations process. The next thing, purification, it is required to get rid of any unwanted components that are coming from the host cell that is being used for vaccine production. So and including serum, whatever is being used, we want to get rid of that. We want only the active ingredient to be part of the vaccine. So that's why purification is very critical, and it is applicable for both live vectored or killed vaccine. Coming to the concentration, this again may, or it again depends on the process, and if at all the antigen content that is required is achieved only by purification, there is no need to do further concentration. If it is not achievable, then depending upon the need, a concentration step is included. So what can QC QA do here? QC QA can do a lot of things. They can check for the sterility. They can look for how much residual BPL is there. So once we add BPL for inactivation, they even look at whether residual BPL is there, and they look at inactivation kinetics. How much of BPL is added, and how much time it takes for the virus to inactivate. So based on these documentation, this goes forward. So formulation and filling, antigen, how much antigen to add. This again, this knowledge comes from the R&D studies, stabilizers, adjuvants, whatever is required, everything is mixed, blended, and is stored until filling, followed by filling the virus. So this go again, what can QC and QA do here? So they can do a lot of things here. They can look for the antigen content in the vial as per the dose that is defined, and they can also look for the sterility of the vaccine that is filled, and storage conditions, storage stability. These are very important things which are required for the manufacturer to prove that even at what temperature, if it is stored at if the stability study is done for 2 to 8 degrees, the vaccine need to be stored, and how long it is stable. So those kind of studies need to be performed by QC, and QA need to support those kind of activities. So coming to the quality control department, there are a lot of branches within quality control. There is chemical department, microbiology, virology, animal house, and environmental health and safety. So these all comes under quality control department. So QC chemical, they do usually water testing, conductivity, pH, heavy metals, chlorine content in the water that is being used for vaccine manufacturing, and they also do antigen testing, DNA content in the vaccine, bovine serum albumin content, and host cell protein content in the vaccine, which there are all limits set for this, so it should match the set limit and the set criteria. So coming to the other part, so they also look at stability of the antigen. They do a lot of calibration studies and method validation studies. QC microbiology, they do environmental monitoring of the production area and the GMP area, which is very important for the manufacture of a sterile product. So this is something that I pulled out from WHO TRS. In this table, you can see that they do look at what are the limits for the microbial content in the air sample, settle plate, contact plate, and glove print. So this they there are some organisms that are used for testing the plates that are exposed in these GMP facilities. So this is very stringent. So they need to even prove that the plates that are exposed in these areas, different areas, grade A, B, C, D, these areas, they are capable of supporting these fungus and bacteria. So these 3 bacteria and 2 of the fungus. So So it's all based on science. All regulatory aspects are based on science. So now coming to the other aspect, QC microbiology also looks into sterility of whatever sample they get, and bacterial endotoxin test, and they also do microbe identification. If at all there are some microbes, they look at what type of microbes are growing on the plate, and is it changing the trend? Is there any different type of microorganism is growing which should not be there? So coming to the QC virology part, they take care of virus titration, which includes tests like plaque assay, TCID, immunocytochemistry for the viruses which are not producing any cytopathic effect on the cells, and hemoglobin for influenza. So these are some of the tests that they do, and apart from that, they also do virus amplification. So if there is an inactivation to prove that the virus is inactivated, for each batch, this kind of tests are required, and they also do identity of the virus, neutralization, and PCR-based testing. So, stage virus particles. In case of ad vectors, these are important things that they do. So, this virus particle, I will explain it to you on the later slides. So adventitious agent test is another important thing that they do to make sure that there is no other virus or bacteria growing in the vaccine samples. They also get involved in cell line and virus characterization, and which is very critical. And they are the ones who bridge between the regulators, QA, and the production people. So they set forth what and all things need to be tested for characterizing the cell line as well as the virus, and they do those tests that are required. Either these things can be outsourced also, but it all depends on how they want to do it. These are some of the tests that you can see here. So this is a typical hemoglobin test that is done for influenza. So if the RBCs are a virus and RBCs are mixed, if virus is present, you can see a mat kind of structure. So since the virus is diluted, as the virus concentration is going down, it's no more capable of suspending the RBC in the solution. So you can see a button formation in this tube, in this 96 well plate. So earlier I showed some uninfected and infected cells, where you can see that the monolayer, this is cell monolayer, and the cell monolayer was damaged in the earlier image. But here, there are some viruses which won't produce visible cytopathic effect. So to quantify them, there are some indirect assays required, where we use antibodies against one of the viral protein which binds to the infected cells, and later we detect that antibody by using a secondary antibody, and count the spots, and then we can estimate the virus content in the samples. This is another department, animal house, which they maintain different animals, mice, rabbits, rats, and hamsters. So it all depends on what animals are being used for what type of test. So they do pyrogen testing in rabbits and animal potency in whatever the vaccine or whatever the standard protocol demands. And preclinical toxicity studies are also done in these animals. And apart from that, animal also takes part in reagent development, like anti-sera against a particular antigen or anti-sera against a particular virus. So these things are done at QC animal house. So coming to the QC EHS, so EHS takes care of the safety aspects of the environment, working environment, and the people who are working in the place. So they also take care of the waste management and keep up to date in the permits that are required for maintaining the EHS. Yeah. So now we'll dive into this viral vectors. Viral vectors, there are, I think now everybody is aware of viral vectors, and a lot of viral vectors have come into the newspaper, and everybody knows about now adenovirus especially. So we'll go deep into adenovirus, but just wanted to uh, let you know that the viral vectors are in place since quite some time. So they were being used for gene therapy, and now it is also this thing. Uh, it's also used for vaccines as well as lab experiments. So it took a lot of time for the vectors to evolve from gene therapy or lab experiments to gene therapy to vaccine. So how are these generated? So if you look at some of these well-known vectors, modified vaccinia ankara or the MVA vector, these are generated by, this is just an example which is taken from a publication. So you can see here wild type vaccinia virus when they are infected in, this is a duck cell line, EF1 cells with a shuttle vector which has the foreign DNA sequence with homologous homologous regions here in the black, and when due to homologous recombination the recombinant virus can be generated. So this virus which are shown here in blue, it comes along with the wild type virus. So slowly like 2-3 multiple times this plaque purification need to be done to get the vector which only contains the this recombinant foreign gene. So this is another vector, vesicular stomatitis virus. If you remember, during this 2016-17 Ebola outbreak in West Africa, there were a lot of vaccine trials that happened. So out of that, VSV-based vector emerged as a good vaccine candidate, and that is approved for human immunization. So this VSV-based vector can be generated by replacing the genome. In its genome, this G protein, it is present on the surface of the virus. So instead of the VSV-G or vesicular stomatitis virus G protein, G gene is replaced with whatever gene we want. So in case of Ebola, it may be just the glycoprotein replaced here. So in corona, for this SARS-CoV-2, spike is replaced here. Spike is added here to its genome. But this alone is not sufficient. You can see here, this is the whole plasmid with the spike gene. It also needs some supporting plasmids which express N, L, P for its generation, and a T7 promoter to kickstart the RNA expression through this plasmid. So upon this thing, serial passage in the cell lines, we can get a virus which no more encodes the VSV-G, but it encodes the spike gene of the SARS coronavirus on it. But it's not the actual virus; it is a vectored virus. So coming to ad vector development, so we have worked extensively on adenovirus vectors, and this is one of the publications that we did for Ebola. And so here we used 293 cells for adenovirus. Usually 293 cells are used. So we have a special cell line called as 293 AICTE cells that was used for generation of adenoviruses, and we have a 2 plasmid system. So we will go into that. So I have split this into 3 parts: generation, characterization, followed by purification and quantification, and then finally immunization and immunogenicity. So so to look into the first part, to generate the adenovirus, 1 minute. Am I audible? I just wanted to know.
Yes, sir. You are perfectly audible.
Sure. Yeah. Yes. So looking at the generation and characterization part, before we do any vaccine or vector vaccine, we plan for that, we need to know which protein or which gene to be considered for making the vector. So here in this case, we I will talk about Ebola. Whatever we have done, we are, so in Ebola, if you see here, this is the Ebola virus structure. It is classified under Filoviridae. So it has a filamentous structure. So here there are a lot of proteins that are structural proteins that are present. Since it is a negative strand virus, it also carries its own polymerase for initial replication. But here you can see a glycoprotein, GP. So this protein is crucial for the Ebola virus to enter the cells, similar to the SARS-CoV-2 spike protein, which is important for it to enter into the cell and infect the cells. So, so these adenovirus vectors, which are made for COVID-19 vaccination, they all express SARC in one or this spike in one or other form. So similarly, for Ebola, this glycoprotein present on its surface is important for its entry into the cell. So now coming to its genome. So if we look at its genome, unfortunately, this glycoprotein is expressed in 3 different forms. Because there is a mRNA editing here, it can in the genome codes for secretory GP, which is expressed in very high concentration. In the second reading frame, due to addition of like 1A here, it leads to the generation of full-length GP, which is present on the surface. So the third form of GP is a shorter than the secretory GP. So they call it SSGP. So the full-length GP is again cleaved into GP1 and GP2 by the host furin, which goes and it trimerizes and forms the part of the virus structure. So if we look deep into this cleavage by host furin, so if we see it, this is how it's present on the surface of the virus. So this is just a monomer form that I'm trying to show here. So it usually exists in a trimer. So you will have 2 fragments of it, GP1 and GP2. So GP1 fragment is the one which is mainly responsible for the entry of the virus into the cell. So we took the sequence from gene bank and added whatever sequences that are required for the expression in adeno expression in the mammalian cells, like Kozak sequence. Then this is signal sequence of the native GP. And as I was stating earlier, so we added a mRNA editing site here. We altered the site so that it only expresses the full-length GP and not the SGP or SSGP. And to the end, we added 2 stop codons to stop the, and apart from that, we also added some restriction sites here. As you can see, ECOR1 and the 5' end, and HIM3 SAL1 restriction enzymes in the 3' end. This is mainly for cloning. So we got synthetic genes of this, and then we subcloned it into, subcloned the GP into adenovirus shuttle vector. By using those restriction sites, which again followed by this generation of the adenovirus. So what we did here is, it's a 2 plasmid system that we have. So we used genomic plasmid, which has the entire adenovirus genome except for this region that you are seeing here. So this is furin. This region is FRT. So so this region, it is from here to here in the shuttle plasmid, and from here to here in the genomic plasmid recombines when they both are transfected into the cell. So the conditions are for generation of advirus in this case is both genomic plasmid and shuttle plasmid should get into the same cell, and both of them should linearize from here, here, and here. These are ITRs, inverted terminal repeats. And after linearization, this region of the genomic plasmid is swapped with this region of the shuttle plasmid. So it also achieves the packaging signal from the shuttle plasmid. Then, when this recombination happens, after this recombination happens, the adenovirus is generated. So we generated control adenovirus using empty plasmid, empty shuttle plasmid, and we generated two Ebola GP expressing adenoviruses by transfecting the Ebola GP containing shuttle plasmid and the genomic plasmid into the 293 cells. We co-transfected and generated the adenovirus. So we did electron microscopy imaging of the recombinant adenoviruses that were generated. So we named these are AD515 is the control adenovirus, and AD-GP72 and 75 are the two different clones of adenovirus which are supposed to express the Ebola glycoprotein. So as you can see here, this is the typical image of adenovirus, and this is the icosahexavalent. So something similar to that we can see in all these electron microscopy images. So this is published already, so you can refer to this publication if you want to get some further details. So inducible expression system, but yeah, this is one more thing that I would like to mention in detail. So this is the whole adenovirus genome, and this is the region where our glycoprotein is introduced, and it has a controlled expression system where the MCMV, murine cytomegalovirus promoter-driven expression of GP is controlled by a LAC operator. So what happens in, we have a cell line called as 293 IQ cells. It expresses high-level LAC repressor. So when we want to produce the vector, when we want to produce the adenovirus vector, we don't want the glycoprotein to express. So we suppress the glycoprotein expression during the production of adenovirus. We have limited amount of cells here. So with that cells, when they are infected with adenovirus, the LAC repressor suppress the expression of the glycoprotein, which is not at all required during adenovirus generation or adenovirus production. On the other hand, if we are using other cell line or like 293 or any other cell line, which does not express the LAC repressor, or let us say if we are being injected with the adenovirus, so we don't express LAC repressor. So what happens is that there will be a high level expression of the virus, this Ebola glycoprotein. So this is how it happens. You can see here in 293 IQ cells, RNA polymerase, it's difficult to bind and express the glycoprotein because LAC repressor is controlling the expression. But when we add IPTG to this, IPTG and LAC repressor, they bind together, and there will be expression of this. So there is a reversal of expression, you can see. But in other cell lines like 0E6 or 293, there is no control on the expression. So there will be a high amount of expression of glycoprotein. So we proved that by doing an experiment where we took cells and we infected them with adenovirus, and 24 hours post infection, we lysed the cells, and we did STS page followed by Western blotting with antibody which binds to the GP2 fragment of the glycoprotein. So here you can see that when we infected 0E6 cells, this is uninfected lane, uninfected 0E6. This is control adenovirus infected lane, and this is GP72 and 75 infected lane. You can see a specific band corresponding to the size expected for GP2 in these two lanes alone. And when and remember that this does not have a control on the expression. So we then went ahead and they did infection of 293 cells where there is again no control on expression. So you can see a good amount of glycoprotein being expressed in these two cases. But when we use the same virus, same treatment for 293 IQ cells, we don't see much of expression of GP. You can see the intensity has gone down drastically. But to reverse it, again, if we add IPTG and treat these 293 IQ cells with IPTG, we expect a reversal of expression. So that is what happened when we treated the 293 IQ cells with IPTG. We could see that the expression levels are similar to what we observed with the 293 cell line alone. So here as a control, we loaded the cell control and we anti-gap DH antibody we used to show that all lanes contain equal amount of sample. Coming to the next part, we did purification of this adenovirus using Q-Cypress beads, and then this is one of the image. You can see here the slowly after purification flow through, we are seeing some loss. Non-specific proteins are gone. Then when we used 0.2 molar NaCl, 0.3 molar NaCl buffer, there is whatever impurities need to go out, they have gone. And please see that it is 9 sample and 37 samples. So in between samples are not there in these images. So 0.4 molar NaCl we used for purification, and you can see here this is, you can see protein band corresponding to the hexon protein of adenovirus. And as a positive control, we used adenovirus positive control from ATCC, American type culture collection. And we also checked for anti-AD5 hexon antibody. We looked for the hexon in these samples, and we can see that in these samples, which are, this is a pooled elution of 0.4 molar NaCl. You can see that the protein bands that are visible here in Silvers in Comasi are in fact hexon. And similarly, the band that is visible in AD5 from the positive control is again hexon. So after concentrating this, we then did immunization and looked for the immunogenicity. So we had 8 groups where this ISI means it's intranasal, subcut, and intranasal. Just for our convenience, I added it intranasal, subcut, intranasal, and SII means subcut, intranasal. So we had 8 groups. These are the 2 different ways we did the immunization, and again same thing, placebo PBS, phosphate buffered saline, control vector, and the 2 GP expressing vectors. So these were used for immunization, and the schedule was like this: on 0 day first dose, 21st day second dose, and 59th day the third dose through the respective routes. On the bleeding, we did on 14, 31, and 67, and we also took a pre-bleed sample, and each dose contained almost 10 to the power 11 virus particles in that. So we wanted to test the immunogenicity. For that, we needed some antigen. Unfortunately, for Ebola is not reported from India, and handling Ebola is not possible in India. So we generated our own ELISA antigens for this purpose by infecting Vero E6 cells with either the control virus or the GP expressing AD vector. And 48 hours post-infection, the cells were lysed. The cell pellets were freeze-thawed 3 times, and then we took the clarified supernatant and coated it and used it as ELISA antigen. So we prepared 3 ELISA antigens here. One using Vero E6 lysate, Another one using control adenovirus infected Viro E6 lysate, and the third one is the ADGP infected Viro E6 lysate. When we used the uninfected Viro E6 lysate and tested the serum samples that we got from the different basically mouse group, what we looked at is more or less there was no all of them where all these bars are very low. So this this was expected. So coming to the all bleeds are looking similar in all the groups. So the next antigen that we used was the control adenovirus infected Viro E6 lysate. So this antigen also does not contain any specific glycoprotein, specific protein in that. So we don't expect any antibody to be present against the vector or any other protein. So accordingly, we did not see any bars going up in these controls as well. But when we use the ADGP Viro E6 infected infected Viro E6 antigen, in this case, we could see that in the groups which received, in the mouse groups which received ADGP clones, the bars are going up in the second dose and third dose. So this happened with both types of immunization regimen, what we followed, that intranasal, subcut, and intranasal, subcut, intranasal, intranasal. In both types, you can see that there is an increase in the response in these two groups. But when we looked at the control group, PBS or this AD515 infected groups, we don't see any response going up. But the question was that we made an adenovirus and we used that same adenovirus to generate an antigen for ELISA. That was we were thinking then what else we can do to generate another glycoprotein from another source. So since nothing was available commercially that time, what we did was we generated a baculovirus, recombinant baculovirus expressing Ebola GP or Ebola HA-VP40. So VP40 is another structural protein which is present on the Ebola. So earlier publications, when we looked at earlier publications, we saw that the VP40 and VP40 and GP, when they are co-expressed, we can see that they form a virus-like particle. So we wanted to exploit this for generating an antigen for the ELISA. So we generated two recombinant baculoviruses, one which express the Ebola glycoprotein, another one which express the HA-VP40. So these two were generated, and we confirmed the generation also. Since we are not having antibody against VP40, we tagged the VP40 with HA tag for it to be detected when it is expressed in these insect cells. And we used SF21 cells for the generating the baculovirus and further expression of the antigen. So SF21 cells are insect cells. They are derived from Spodoptera fujiperda, and there are two types of these cells available: SF9 and SF21. There is some variation in the doubling time of these cells, but we used SF21 cells, and we infected them with either baculovirus expressing GP or baculovirus expressing HAVP40, and 72 hours post-infection, these were lysed, and we did SG stage and Western blot to confirm that they are expressing GP. You can see here the GP expression using the antibody which was used earlier. So, and we also showed that it is the baculovirus HAVP40 is expressing the VP40 by using anti-HA antibody. And we also wanted to test whether these two are associated with each other by biochemical means. So if you can see here, this is VP40 here. These bead kind of structures that you are seeing here in the Ebola virus. These are VP40, and if you take a look closer, these are somehow associated with the glycoprotein. So we wanted to prove it by doing immunosuppression. So for that purpose, we again infected these cells, SF21 cells, with baculovirus expressing GP and HAVP40. And 72 hours post-infection, we did ultracentrifugation and over 20% sucrose cushion. And expect to see that the pellet contains both HA and HAVP40 and GP associated with each other. So for that, we did the immunosuppression. We used two antibodies here, anti-HA to pull down the VP40 and detect both VP40 and GP in those samples. We also did vice versa, where we used anti-GP antibody to pull down the GP expressed by baculovirus, and then looked at whether we can detect both GP and VP40 in the other case. So if GP and VP40, HAVP40 are associated with each other and forming a structure, we will call it as higher order structure. And if we pull this also should get pulled, and we should be able to detect the other protein. If we are pulling this, we should be able to see the other protein associated with it. So we did a Western blot, and you can see here that GP, when we pulled the lysate using anti-GP and anti-GP antibody, and we could detect the GP presence in the sample. This is obvious, but when we did anti-HA immunoprecipitation, we could see the presence of GP here. These are very faint bands, but they are we can make out that they are associated. So this shows that the VP40, HAVP40 is associated with GP. We did the vice versa also. So here you can see that the bands that are visible in the green box, it indicates that the anti-HA antibody detected HA tagged protein here, which is corresponding to the size of HAVP40. This, when we do anti-GP immunoprecipitation, there also this HAVP40 was visible. So this indicates that these two are associated with each other in some structure. We did not go for electron microscopy for these structures to prove ahead. So since our idea was to use it as an ELISA antigen, we went ahead to use it as an ELISA antigen. So we used two ELISA antigens. One is baculovirus expressed glycoprotein alone, or the higher order structure. So these two were used as ELISA antigens, apart from the mammalian derived ELISA antigens that were used earlier, what I showed earlier. So when we use baculo-GP antigen, we could see that that there is again a spike in the response in the groups which received the AD GP72 or the AD GP75 vector. While the response is low in PBS and AD5 control adenovirus group. This is true across both the immunization regimen that was followed, and we can see a corresponding increase in the other in the last two groups as well. So similarly, when we used baculo higher order structure antigen as the ELISA antigen, we could see that a similar spike in the response were observed in the second and third dose. After the second and third dose, and you can see the increase in the spars in only the specific groups. So with this, I would like to conclude my presentation, and as acknowledgements, I would like to thank Dr. Krishna Ella, CMD of Bharat Biotech, who is also the founder of Ella Foundation and the director, and Dr. Nagendra R. Hegde from NIAB. He has moved to NIAB. He is my mentor, and he is currently the director in charge there, National Institute of Animal Biotechnology at Hyderabad, and our Ella Foundation team members. So this is our team now. Yeah, thank you very much, and I would like to happy to answer questions.
Thank you so much, sir. There is one question in chat box from respected Dr. Vandana Patrao, ma'am. May I read the question for you, sir, please?
Yeah, please.
Any specific reason why ultracentrifugation is performed on sucrose cushion? Is it a routine practice?
For ultracentrifugation.
Yes.
Okay, so it all depends on how what is our need, and here the idea was this is regarding that ultracentrifugation for baculovirus, isn't it? That is what she is asked. Okay.
Yes,
so
yes.
Yeah. It all depends on what is our need. So here, if we are trying to see, like we are generating a higher order structure, let us say, or let us say adenovirus, whatever can penetrate into the sucrose, individual proteins cannot penetrate. If we are using 20% or 30% sucrose or more than that, when we do ultracentrifugation, these individual proteins cannot penetrate the sucrose because of its density. So only a compact structure can penetrate into that. So when we are trying to generate a higher order structure or a virus-like particle, which is expected to be compact because of the expression of two proteins which are associated with each other, we expect it to cross that sucrose, dense sucrose, and pellet down. So because a lot of other proteins are also there which are coming from the insect cells, we don't want that to be like part of this analysis. That is the reason why ultracentrifugation was used.
Thank you so much, sir. I request participants to unmute themselves and you can ask the questions.
Sir, in case of Covaxin, you felt to go for the conventional mean of developing viral vaccine, while there are advanced techniques been available, like how it has come up with Covishield. What was the earlier thought in the development?
I don't think I should talk about this. Already a lot of things are there in the media. So always conventional vaccine is better. We prefer to go with that, and that is one of the reasons. I'll put it that way.
Okay.
So was my presentation, I hope I did not bore people here with my presentation.
No, the thing is what people are from pharma background, right? They're not that worked with the microbiology.
Yeah, that is true. But we see a lot of people from pharma background in in vaccine companies nowadays. So
this is the aim for it, no? So that they can think of entering into the biological science.
Yeah.
Thank you so much, sir. Actually, because you accepted our invite in very short period of time. At 11th hour we called you. Somehow we could reach to you very late.
Yeah, yeah, it's all because of Dr. Amit Mishra. Actually, this has happened. Or
Correct.
else
Correct.
because he introduced and then I thought, okay, this is
Yeah.
right. That is why this is.
So that's it. I
Yeah,
think
yes.
we can take.
Yes. Thank you, dear delegates, for your queries and Deepak sir for answering them aptly. I would like to take this opportunity to express my sincere thanks to our speaker Deepak sir. We truly appreciate your dedication for sharing your valuable experience with us. Thank you, sir. Sir has highlighted on regulatory roadmap for vaccine development as well as production of the vaccines. You made us understand concept of upstream processes as well as the downstream processes very clearly, as well as the role of the QA/QC in every step of the vaccine development, quality control of the animal house, as well as adenovirus vector development, inducible expression system. So thank you so much, sir. We would like to take this opportunity to felicitate our guest speaker with our appreciation certificate. May I request Dr. John sir to do the honor, please?
Sir, thanks a lot for sharing your
Thank you, thank
expertise
you,
on the viral vaccines production. Thank you.
thank you very much.
Thank you so much.
Thank you.
Thank you, sir. I request all the participants to put your video mode on for a snapshot with our guest speaker. Sawan sir.
Yes, sir. Done, sir.
Thank you, sir. Thank you, delegates. Thank you, sir. Deepak sir.
Thank you, thank you.
Thank you, Dr. Deepak. Thanks a lot.
So I'll log out now. Yeah, thanks.
Bye.
Thank you, everyone.
But sir, we are going to start at sharp 2:30, right?
Yes, sir.
Yeah.
So there will be a lunch break of half an hour. So we'll resume our next session sharply at 2:30 p.m. I repeat, we are going to resume our next session sharply at 2:30 p.m. Thank you, delegates.